Hello everyone and welcome back to Neuropsychology. So we have covered a lot in the previous videos and today we're going to start covering chapter 5 of the book Fundamentals of Human Neuropsychology by Kolb and Wishaw. So in previous videos we covered a lot, so here are just some bullet points of what we covered, but we covered way more than this. So we talked about cell anatomy, how proteins are made, resting potential, graded potentials, IPSPs, EPSPs, action potentials, and myelination, and how that causes saltatory conduction. So I know this is a lot, but we covered even more. So if you skipped some of them or missed some of them, please go back and watch those. And then in this video, we are going to talk about neurotransmitters. What are neurotransmitters? The structure of a synapse, how neurons talk to one another, so how they convey information from one cell to the next one, and all the different steps in neurotransmission. Okay, so we all know by now that the dendrites are the parts of the neuron that get the nerve impulses from axons of other neurons. So this is technically a little recap. So these nerve impulses can then result into excitatory postsynaptic potentials, EPSPs, or inhibitory postsynaptic potentials, which we call IPSPs. So when an EPSP occurs, the cell membrane depolarizes, making the inside membrane or the inside of the cell a little bit more positive compared to the outside of the cell. So this is excitatory, and then it makes the cell more likely to fire an action potential. So when an IPSP occurs, which is the inhibitory postsynaptic potential, the cell membrane hyperpolarizes, making the inside of the cell a little bit more negative compared to the outside. So it pushes it away from the threshold, and therefore it's inhibitory, and it will make the cell less likely to fire an action potential. So in the axon hillock, all these EPSPs and IPSPs, they're summed up through temporal and spatial summation. And if it reaches the threshold so if it goes from negative 70 millivolts, which is the resting membrane potential, to negative 50 millivolts, which is the threshold, then an action potential will occur. And action potentials are all or nothing. So whenever the threshold is met, the whole thing will happen. The whole action potential will happen. Okay, so let's talk about the synapse. So there are three main parts of a synapse. So the first things first, at the end of the axon, the cell before the synaptic cleft is the presynaptic neuron. So this is the cell with the axon and with the axon terminal. So the axon terminal is the end of the axon. So right here in this picture, you can see that this is the end of the axon. So this is a zoomed in one from here. Here you see a beautiful little cell with all the dendrites in the cell body, and then this axon going down right here. And then here you have a dendrite of the postsynaptic neuron. So this little piece could be something like this, but from a different cell. So this area right here, this is the end of the neuron, and at the end of this, we call this the presynaptic membrane. So this membrane is the presynaptic membrane, which makes sense because this whole thing together is a synapse. So presynapse is the presynaptic membrane. So this membrane consists of a lot of protein molecules, and most of these are channels, some pumps, and also some are receptor sites. Okay, so then we have the synaptic cleft, which you can see right here. It is a space between the synapse of one cell and the dendrite of another cell. So then right here you have the dendrite of the next cell. So on the tips of these dendritic spines, is the postsynaptic membrane. So don't get confused with pre and postsynaptic membranes. They technically make sense. Pre means before, post means after. So postsynaptic membrane is the membrane that comes after the synaptic cleft. So this area consists of many protein molecules, and these are very specialized for receiving chemical messages. So these chemical messages are what we call neurotransmitters, and we are going to talk about that a lot in this video. So going back to the presynaptic axon terminal, there are many other specialized structures, including mitochondria, which you can see here in pink, which are needed for energy. And then there are synaptic vesicles, which you can see here. So synaptic vesicles technically are just big sacs that kind of hold neurotransmitters. And then you have granular cells, which you can see here. They're big compartments that just store these vesicles. And then you also have these tubuli right here, and they technically give the synaptic terminal its shape. So in some axon terminals, there are these storage granules. I can't pronounce that word, sorry. But some axons have these, but some axons don't have those. Okay, here you see the same picture, but with all the labels. I just didn't want to overwhelm you, and I wanted to talk about it first before you started looking at all these labels. So let's look on the left side first. So here on the left, you see an electron photomicrograph of a synapse. So look how cool these colors are. So you have a bunch of cells here. So here you have the presynaptic membrane, and here you have all these little vesicles, and these blue vesicles, they hold the neurotransmitter. So here you have the cleft, and here you have the postsynaptic membrane. So this is the axon, this is the dendrite, and this axon is gonna let all these chemicals fuse over here, diffuse over. They're gonna attach to the side, and they're gonna send messages in here. The neurotransmitters themselves don't go in, but they just send a message and then they move on. And then here on the right, you see the same picture that we just went over. So here you have all the microtubules, which are important for transport and also for creating the shape. You have the mitochondria, which are important for providing energy to the cell. Synaptic vesicles, they have the neurotransmitters. The granular cells, which hold synaptic vesicles, that then again hold the neurotransmitters. So this brownish looking thing is the presynaptic membrane. Here you have the synaptic cleft, and here you have the postsynaptic membrane. So here you see these yellow little things, those are channels, and they can also be receptors. So these little tiny neurotransmitters, which you can see all these little dots, they can diffuse over and bind to these receptors, and then this receptor will send out a message to the cell. So what I want you to see here is that these neurotransmitters, they're in these vesicles, and these vesicles kind of float over, and then they fuse with the presynaptic membrane, and they kind of just pump out all these neurotransmitters. Okay, so when an action potential goes down the axon and reaches the end, so the axon terminal, the vesicles that contain these neurotransmitters will fuse with the presynaptic membrane, as I just said, and the neurotransmitters will be released into the synaptic cleft. So these neurotransmitters will diffuse over the synaptic cleft, and they will travel to the receptors on the membrane of the postsynaptic cell, which is the dendrite. So that then these neurotransmitters will bind to receptors, and these receptors are proteins embedded in the postsynaptic membrane. And then whenever that happens, this receptor will deliver a message to the cell. So this message could be an EPSP, or an IPSP, greater potential, or it can cause different kind of messages. But for now, I just want you to remember that after an action potential, the neurotransmitter gets released into the synaptic cleft, it diffuses over the cleft, binds to the receptor of the postsynaptic cell membrane, delivers a message, and then after that, whenever it did its job, it gets taken away in some form, which we're going to talk about in a couple slides. Okay, so what is a neurotransmitter? So a neurotransmitter is a tiny chemical molecule that carries a message across the synapse. So there are many types of neurotransmitters, and you probably heard of a bunch. An example of a neurotransmitter is dopamine, or for example, serotonin. So neurotransmitters are released from one axon to the dendrite of a neuron. They diffuse over the synaptic cleft and they will attach to the receptor on the dendrite. So I know I'm repeating myself a lot, but I know this can get confusing. So I hope if I repeat myself enough, you will memorize this by the end of this video. So when they connect to the receptor on the postsynaptic cell, they might excite the neuron through an EPSP, or they can inhibit the neuron through an IPSP, or they can create other reactions. Okay, so there are different types of neurons and different types of neurotransmitters, and groups of neurons that release a certain type of chemical neurotransmitters are often named after that neurotransmitter. So you have 3 examples right here. So for example, a neuron that releases acetylcholine are called cholinergic neurons. Neurons that release epinephrine are called epinephrine neurons. And neurons that release noradrenaline are called noradrenergic neurons. So this is what they look like, but we're not going to go into further detail of these. Okay, so before we go further, we need to talk about the specific steps in neurotransmission. So I know there's a lot of text on the screen, so let's just go through this one by one. So the first step is transmitter synthesis and storage. So neurotransmitters are created in the cell's DNA, or the building blocks for these neurotransmitters are imported and stored in the cell. So neurotransmitters are then gathered in the synaptic vesicles, and those can also be collected in these storage granules. So here you see in pink these neurotransmitters, here you see the vesicles, and here you see these storage granules. Okay, so then they will attach 2 little microfilaments in the terminal button, and eventually they're attached to the presynaptic membrane, and they will kind of get pushed out. So this is the step of neurotransmitter release. So how neurotransmitters are released depends on the action potential. So whenever the action potential comes all the way to the end of the axon, this will open up calcium channels in the cell membrane, and then a lot of calcium will go into the axon terminal. So the incoming calcium will bind to what is called calmodulin. We're not going to go into detail of this whole process, but because of these calcium ions binding to the calmodulin, this triggers the release of neurotransmitters into the synaptic cleft through exocytosis. So this is what you see here, is where these vesicles kind of fuse into the membrane and push all the neurotransmitters out. So the third step is receptor action. So after all these little neurons, they kind of diffuse over and float around, they will go to the other cell, they will attach to the receptor, and And that is our job. All they have to do is go over, bind to the receptor, and that way, through the binding, they will deliver their message. So whenever they did their job, they're not actually going to go into the cell, but instead, after they bind it to the receptor and did their job, they will let go, and then they will either be sucked back into the terminal or broken down, or get inactivated. So they can actually also be sucked up by the presynaptic cell, so the axon, so they can be reused. Yay, recycling! Okay, so the first step, we're going to go over it again. First step is synthesis. So neurotransmitters normally are made in the DNA, but sometimes the building blocks are stored in the cell itself. Then so whenever the action potential comes down, calcium will go into the cell. This eventually will push the neurotransmitters out of the axon. The neurotransmitters will diffuse over, then they will bind to the receptors, they will deliver their message, and then they will either get taken up by the cell or broken down, or they diffuse out of the cleft. So they will get either reused or inactivated. Okay, so what happens when a neurotransmitter binds to the receptor? So whenever you see NT, I'm talking about neurotransmitter. So there are a bunch of things that can happen. So the first thing that can happen is they can depolarize the postsynaptic membrane, which means they can give a little EPSP to the dendrite. And if you have enough of those EPSPs, you all know what will happen. The postsynaptic cell will eventually also fire an action potential. So when they depolarize the postsynaptic membrane, they will make the next cell more likely to fire an action potential. They can also do the opposite, and they can inhibit the next cell. So they can hyperpolarize the postsynaptic membrane. So they make the inside of the next cell a little bit more negative, which takes it further away from threshold, and therefore this cell is less likely to fire an action potential. They can also initiate other chemical reactions that can be excitatory or inhibitory. We're not going to go into the detail of those. They can also generate new synapses, or if there isn't enough of the, or if there's too much of the neurotransmitter, they can take receptors away, so they can technically also change the cell. Okay, so let's go to the last step. So after a neurotransmitter has done its work, it is removed very quickly from the receptor and then also from the synaptic cleft, so there's more space for new messages that are sent by the presynaptic neuron. So neurotransmitter deactivation takes place in at least 4 ways. So the neurotransmitter can be deactivated through diffusion. So here, the neurotransmitter can technically float away or diffuse away from the synaptic cleft. Next, the neurotransmitter can also be deactivated through degradation. So here, enzymes come in and they break it down so it cannot send any more messages. So this is kind of like Pac-Man, where Pac-Man just eats the little ghosts. And a third way is a neurotransmitter can be taken back into the presynaptic axon, so it can be reused, yay recycling, or sometimes glial cells can take up some of the neurotransmitters and then store them for later reuse in the axon. So this was the end of this video, and I will see you guys back soon for the next video.